Hi, Dr. Kaufer. How are you?
Hi,
Thanks for coming.
thanks. I just sent you an email. Thank you for resending this.
Oh, yeah, sure. Let's wait for a few minutes and give people time to arrive, and then I'll introduce you, and then, yeah, the stage is yours. So, thanks for coming. And please meet Dr. Mariam, and yeah, she will also be co-hosting, and Cecilia, yeah, yeah, say hi to Dr.
Great.
Kaufer.
Hi, Dr. Kaufer. My name is Cecilia. It's nice to meet you. I'm excited to hear your talk. Yeah.
Thank you. Nice to meet you. And nice
Hey,
to meet
Dr. Kaufer. It's really lovely to meet you as well, and looking forward to speaking with you.
Great. Hi, Dr. Mariam. It must be very late for you guys.
Oh, yeah, it's 10 p.m. So here in New York, I think for you it's like 3 hours earlier, right? It's 7 p.m.
Yep. Yep. It's going to be 7 p.m. Yep.
Right. So, do we want to give it another few minutes before we get started, or
Yeah, the event is announced for 10 p.m. I just yeah, wanted to make sure uh yeah, the room is open and people have time to arrive. So yeah, let's wait a few minutes.
Yeah, that's great.
Yeah, I'm pinging people in now. Yeah, I think this is going to be a wonderful discussion.
Yeah, as I'm watching the Russia-Ukraine standoff, UN Security Council live stream, I'm like, oh, anxiety. I'm like, oh, I wonder if anything here is of interest there, because it's a very anxiety-inducing situation. So interesting to see. So we're talking about anxiety, so very appropriate, very timely.
Yeah. Welcome, everybody. Welcome. We're going to start in a couple of minutes. So just hang tight. Yeah, we're starting at 10 p.m. So in just 2 minutes.
Yeah, Eric, I was a child when living in Germany when the Cold War was still and the wall was still up. So yeah, it's not a very comfortable situation. I don't know. I think it's very nerve wracking, but we'll see what comes out of it. Right? We don't have much say, I would say.
Yeah, we need somebody like Mr. Gorbachev tear down that wall. It's weird how like an actor could have prevented something. Although I suspect some sort of interaction with Trump will probably play into effect here, because Putin seems to be playing multi-dimensional chess, and sometimes it looks like our side is playing checkers. So T-1 minute.
Yeah. And I'm just, trying to ping more people in. Katarina, are you able to share it on Twitter?
Yep, I shared it on Twitter, and yeah, it will probably people will. Oh, right. I have one more quick thing to do, and then I'll.
Yep, sharing it too. And also sharing it on Clubhouse. All right, it's 10 p.m.
Oh, hi, Dr. Olu. Thanks for coming. I appreciate it. Yeah, meet Dr. Daniela Kaufer.
We actually overlapped for a little bit. Um, I was graduating from the Sapolsky lab when I think she joined way back when.
Yeah, that's true. I remember.
Good seeing you again.
You were working on 11 beta HSD.
That's right. That's right. Good memory.
Nice, dude. Yes. How have you been?
I've been good. I've been good. Really excited to dive more into this paper. Just nice to see really good translational work.
Thank you.
Wow, that's great. The world is small. Dr. Olu, he has been, coming to many of our rooms. We co-hosted rooms together. So yeah, it's nice that you get to see or meet each other again this way. That's great. Okay, welcome to Science Society, everyone. We are very honored to have our guest speaker, Dr. Daniela Kaufer, and she will be talking about her latest research that has had quite some attention in the news world, about her study of showing increased myelin linked to anxiety and PTSD. And I will introduce you. Um, Professor Daniela Kaufer is the class of 1943 Memorial Chair Professor of Integrative Biology and Helen Wills Neuroscience Institute and the Associate Dean of Biological Sciences at UC Berkeley. Professor Kaufer received her PhD in molecular neuroscience from the Hebrew University in Jerusalem, and she was a postdoctoral fellow at the Department of Biological Sciences and Neurosurgery at Stanford University. Kaufer's lab is studying how does the brain deal with perturbations like stress and brain injury, and what are the mechanisms underlying trajectories of resilience and vulnerability throughout life and into aging. The research had been published in top journals including Nature, Nature Medicine, Science, Translational Medicine, Nature Neuroscience, Annals of Neurology, eLife, and Molecular Psychiatry. Professor Kaufer is a recipient of the National Institute of Mental Health Directors New Innovator Award, BRAINS, NARCET Young and Independent Investigator Awards, Bakar Entrepreneurship Fellowship, and the Archer Award for Innovation in Aging Research. So, yeah, we are very honored to have you, and thanks for coming, and the stage is yours.
Hi, everyone. Really excited to be here. I'm still learning the ropes of this very new platform to me. So I hope it goes easily. And I want to say that I would love to have a conversation more than anything else. So I could just start by introducing a little bit about what the lab does and what this research was, and really welcome to have this as a conversation and answer any questions and any particular angle that's of interest to people. To me, stories are always more interesting, and then the path to the finding itself, and my path in science and so on. So anything is open, and feel free to just jump in with questions. Um, so yeah, I've been at Berkeley now for almost 20 years. Been at Stanford before as a postdoctoral fellow, and since very early in my career, uh, I was interested, got to be interested in the effects that stress has on the brain. My my road to it was an interesting one, and sort of accidental, I would say. But I started doing something else in grad school, and it was right after the first Gulf War. And the interesting Gulf War syndrome came about. There was this question about soldiers coming home and having responses that were not anticipated before. And in a way, this medical mystery kind of paved the path of the rest of my career in research and in science, finding always translational stuff. So, threading the balance between patients and mysteries that we find there, things that don't make sense or we don't understand the mechanism to, going back to animal models and finding mechanisms, and going back to patients and seeing whether what we found was relevant there. And we do that in a lot of different angles, but the big umbrella to this is brain plasticity. I think the big topic is how the brain reacts to things that change, and stress is one of them. Another part of my lab is working on traumatic brain injuries, and another part of the lab is working on aging, which is another situation where brains change. So we're very focused on plasticity and things that change in the brain and what follows all sorts of injury and insults. And in all those different scenarios, so traumatic brain injury, we were studying football players, and we have animal models of that. In stress, we were studying, in this case, veterans and animal models of an exposure to psychological trauma. In aging, in all of those cases, the theme that continuously repeats is this interindividual variability, is the fact that even in the animal models, when we expose a group of animals that's very homogeneous, they're genetically almost identical, they've had the same life history in the animal facility, so it's a very homogeneous group as opposed to patients or veterans, let's say. Yet following that event in a big group of animals, we will see a lot of different outcomes. You will see animals that actually thrive and show no behavioral effect when they're exposed to trauma. You will see others that are actually doing better. We've had papers in the past where we were asking how stress actually makes some brains even better. And you will have others that show effects downstream of that effect that are behavioral fingerprinting, if you want, of the trauma. And we started to ask why. Why is it? What makes one brain different than another, such that they're reacting so differently? And this is a project that follows my lab from the very beginning of it. In fact, the very first steps of asking similar questions started in the Sapolsky lab at Stanford when I was a postdoc. And we were studying their neural stem cells and seeing that those very particular stem cell population in one area of the brain that is involved in learning and memory, the hippocampus, that group of cells is affected by stress. And it's affected by stress by different ways. Some moderate stress actually makes us produce more of those cells and, in a way, prepares the brain for the next stressor to come around. And more severe or more chronic stress would actually hamper the activity of those stem cells. They proliferate less, they make less of the relevant neurons. And then we got stuck on the question, well, what happens to them? So we see less of those new neurons being born, so they must be dying. So we looked for dead cells and we couldn't find them. Okay, if they're not dying, maybe they're like stuck in some stem cell limbo, and they weren't. And so we kept on looking, and in another part of the lab where we were looking at another phenomena of epilepsy and traumatic brain injury, we found other types of cells, not neurons, that seemed very important to what we were studying there. And those cells are called glia. They were believed for many years to be support cells of the support system of the brain, but they're now more and more known as something that actually contributes to brain function. And in this case as well, when we were looking at those cells, turns out that those stem cells were actually changing their fate, and instead of becoming neurons, they were becoming glial cells. And they were becoming a type of glia that's called oligodendrocytes, and that's a type of cell that produces the insulation that is around neuron connections. So neurons, the neural cells connect to one another when they're sending this long connections, and those connections are like electrical wires. And the electrical wires have a type of insulation around them. You can imagine sort of the plastic insulation that you would see around an electrical wire in your house. And this plastic, if you will, is called myelin. And the cells that we were finding that all of a sudden were accumulating in stress, in chronic stress or severe stress in some of the animals, were cells that were producing this myelin, and they were producing it in the wrong place and the wrong time. So they were enhancing connection between cells that you wouldn't necessarily expect to see. And we've been following the story for almost the last decade. And this paper now is the last foray in that. And it was a fantastic work by a graduate student in my lab, Kim Long, who started to really probe into, well, what does it mean to have those cells, to have them being produced? She did very deep phenotyping of animals that were exposed to stress and was able to find connections between which animals actually had this myelin overproduction and which ones showed sensitivity to the stress. And even more so, she found that the area of the brain in which we're seeing this overproduction of myelin is related to what symptoms they might show. So there were animals that were showing avoidance behavior, and those were producing this myelin in one aberrant myelin production in one part of the brain. And there were other animals that were showing problems in their fear memory, and they were showing in another brain area. And other groups of animals that had startle response, and they had another brain area. So that was very interesting by itself. We then connected with a group, Dr. Tom Nalen and Dr. Linda Chow at the VA in UCSF. And they did an imaging study in which they took veterans and were trying to ask by MRI imaging of their brain if we could see any proxy of that in the brains of the veterans. And that was the real surprise. That was the most amazing thing to me, because you wouldn't really expect necessarily that our brain is so similar to the rat brain, but it was so similar. Not only was it very similar in the sense that we saw this response, but the areas were connected to the same types of symptoms. So veterans that were showing a lot of avoidance behaviors had hyperproduction of myelin that could be detected by MRI in the same area of the brain where we saw that in the rats. So avoidance, fear memory, and so on were co-localized. The last thing that we did in this paper was something that really highlights one need of animal models is when we're doing this kind of everything that I described so far in humans and in rats is a correlation, but it doesn't really tell us about causation. So we know that those things are correlated. That does not mean that that one thing actually causes each other. They could be epiphenomenons that are arising together. And so we started asking. There's a lot of work still to do, but the first thing that we did, first step towards that, was that we created a viral vector. We created a virus that overexpresses a transcription factor that can push cells to create more oligodendrocytes and more myelin in particular brain areas. And when doing that, we could mimic what would be the effect of stress. And we saw that the animals that were given this viral vector and now overproduced in a particular brain area, we actually saw an increase in avoidance behavior as if they were exposed to stress. So we know that this is sufficient to recapitulate this effect. So that's the paper that we're talking about today. But I'm going to open it up for questions here, and very happy to talk about this paper, other papers, anything that you guys might have an interest in.
Wow, wow. Thank you, Dr. Kaufer. That is quite an amazing story. I'm really glad you're here once again, and I'm happy to hear it. I've got questions of my own, but I'll let it go to the floor first, unless Katarina or Sisi Rahim want to have something to say or something to add.
No, no. I also have questions of my own. So yeah, I think we should maybe just, go through the mods and then go into the audience maybe.
Yeah, sure.
Katarina, do you have anything?
Um, yeah, I have some questions, but maybe they're more specific for later. So go ahead, and then I'll ask later once we have time. But yeah, please go ahead.
Okay. So the question that I had was the areas of the brain that showed the glia, the myelin, um, were they random or were they somehow connected to the stressor that was introduced? Does that make sense?
Yeah, that makes a lot of sense. That's a great question. So we specifically chose areas that we know are part of the stress circuitry. There are areas of the brain that are very well known to respond to stress. One of them is the hippocampus, and we chose about I think there were 10 or 15 sub regions of the hippocampus that we looked at. Another area is the amygdala. So that's an area that responds um, to emotional stimuli, to threat, to fear. It's well known for that, and it's part of the stress circuitry. Another area was the prefrontal cortex. It didn't make it into this paper. It will be in a different paper, but those are very well documented um, areas for this stress circuitry. We have seen a big difference here in the amygdala and in some subregions of the hippocampus that are known to be important for learning, for memory, for learning, for navigation. There's another student in my lab right now that is doing a different kind, and this was all in the same stressor. And the stressor is a stressor that is relevant to the life of the animals. They get to smell the urine of a fox. So this is a predator for them, and it's etiologically relevant. And while they smell the urine of a fox, they are restrained, so they cannot run away. So so physically there is nothing stressful about that, but psychologically it's very stressful for them. Now there's another student in my lab that's doing a different kind of stressor, a social relevant stressor, and she's seeing other brain areas that seem to invoke that response. So it definitely seemed to be stressor relevant as well.
Thank you so much, Dr. Kaufer, for sharing that. All right, Dr. Marion, did you have anything?
Yes, I did. So, I was wondering, and I'm sorry if I missed this in your talk, but did you do any experiments where you exposed any of the animals on like being tested to prolonged amounts of time? And if so, what were the stress, what was the glial contact in their brains? Was it similar to the ones who had received like short-term stress or maybe born under stressful conditions, anything like that?
Yeah, yeah, great question. So this paper that we're discussing today, that was done with an acute stressor, meaning this whole effect comes from 3 hours of exposure to the fox urine. So it's a single event. We thought that was a good place to start, um, to simulate a traumatic event, that you're exposed to one traumatic event. So that was the one time that we looked at it with a very, very short stressor. Right now, in the lab, there are other other much more prolonged stressors that people are looking at. But I don't know yet the answer to what would be the effects there. One postdoc is looking at as a social interaction that takes about 14 days as it occurs, and it's sort of mimicking a situation of a bully. There's an exposure to a bully mouse that they're exposed to every day for 10 days, and then there's another 4 days in which they can smell that mouse but they're not exposed to it. So that's a much more prolonged one. And then a much more prolonged one is an experiment that another graduate student in the lab is doing, looking at you will maybe recognize that from something that we've all been going through in the last few years, uh, social isolation. So it's prolonged social isolation, and then looking at how and specifically we did that in older ages and seeing, does that change aging in the brain as well. So looking at selective vulnerability that comes from social isolation and aging.
Wow, that's really cool. So I'm looking forward to, hearing the outcome of that research as well. So I'll be keeping an eye out. Thank you, Dr. Kaufer.
Yeah, yeah, me
I
too.
think
It feels like we're living that experiment on humans, right?
yes, it feels a little bit all too real. Um,
Yeah.
but it would still be good to know some sort of answer as things, I don't know, as the pandemic progresses or yeah. Okay. Thank you very much. Dr. Olu, would you like to go
Sure.
next?
And congratulations again on a really fascinating paper. As somebody who started off in the Sapolsky lab and is now doing neuroimaging work, I feel like your paper is like a career path, is my career path in a paper. Um, so I have a couple of questions along those lines. So starting off with the oligodendrocytes and the enhancement that you saw in association with the sort of more anxious and avoidant behavior, was that also associated with sort of enhanced neuronal function in the hippocampus and the amygdala?
Yeah. Um, so we haven't looked. I don't have the answer for that yet, I would say. When we started with neurogenesis, it was easier because we were tagging the newborn neurons, and then you could follow them and see their activation. And here, the question is technically, I would say more challenging, is if you have more production of oligodendrocytes in an area, how do you follow the actual cells that this new myelin attaches to? And there's a student in my lab right now who's trying to ask this question while using transgenic animals, where she will record engrams of cells, so tagging cells that are active in the stress situation with a fast trap. And then you come back to those cells later on by reactivating and asking whether you see differences in their myelin and just recording from them. Um, so there's a postdoc who's trying to do that now. It's really it is much more challenging than we thought it would be. But I'm hoping that we'll have an answer to that. But that's exactly I think that's exactly the next step. Is now we have a phenomena, but we really need to understand what it's doing to the neurons around it to know what's the meaning of that. What's the functional meaning? There's a big gap there between the behavior and the cellular phenomena that we recorded.
Yeah, because the other thing I think about is how, an increase in white matter might also be associated with inflammation in a particular brain region. And so it'd be interesting to see what are sort of the functional consequences of this increase in myelination. And then related to the human neuroimaging study, I noticed that the sort of control region that was used is the corpus callosum, which is a major white matter fiber tract in the brain. But you're comparing that to myelin in gray matter region. So I was wondering if your translational collaborators also looked at gray matter myelination outside of the hippocampus and amygdala? And were those regions also not correlated with like the PTSD symptoms and subscales?
Yeah. So, the corpus callosum, you might imagine, did not start as a control when we actually were expecting that we will see a difference in white matter. And that was a big surprise that we're not seeing the effect in white matter. Not to say that it doesn't exist, but with the way that we were measuring it, we didn't see an effect in white matter. Linda Chow was the neuroradiologist who did all of that study, and this is a subset of patients that she looked at, and this is a subset of, or subjects, I should say, not just patients, but this is a subset of the subject and subset of the brain areas. And she has a bigger paper, I think, that will come out eventually that has um, triple the amount of the subjects and a lot more brain areas that she was looking at. Those the hippocampus and the amygdala and seem to be the most major ones that show a difference, and then she also has some differences in prefrontal cortex.
So yeah, I'll be keeping an eye out for that paper. I would be very curious to see if she looks or looked at the uncinate fasciculus, which is another major white matter fiber tract that connects the hippocampus and the amygdala to the medial prefrontal cortex. And but in a lot of clinical studies, we see a decrease in white matter integrity in that particular fiber tract in patients compared to controls, and it often correlates with symptom severity. So I'd be very
Yeah,
fascinated to see
so
what she gets.
so I should say we have now from the social defeat study, we have also a decrease in the in um, connecting white matter track, whether it's the same or not, but connecting white matter track between entorhinal cortex and hippocampus, and possibly hippocampus and amygdala. And there is a decrease there in MVP staining, for example. So what I want to suggest is let's connect by email and
Yeah, definitely.
bring you into the study and write the next one.
There we go. Yeah, I'd be happy to.
That sounds really great. Thank you so much, Dr. Kaufer and Dr. Olu. That discussion was really amazing. Thank you. So yeah, we're coming up on the half hour mark, and I just wanted to do a soft a reset of the room. Hello, everybody, and welcome. This is the Science Society. It's a club where scientists and just everyone can meet and share ideas in the pursuit of knowledge. So please click the house at the top and join our club. We have wonderful rooms coming up this week. There's one next, that's going to be about computational structural genomics of pathogens. It's going to be really interesting. So yeah, thank you so much, everybody, and thank you, Dr. Culver. So we're going to go on next to Brandon. Hi, Brandon. Do you have any questions for Dr. Culver?
Nothing. I don't know if it's related, but just I was always curious because I learned that I guess cortisol, the stress hormone, is always can make the brain portion of the brain more sensitive to dopamine. I just thought that was interesting, and if you had any information on how that works as far as people being addicted to stress or feeling like, I guess, if it makes it, you sensitive to dopamine, how that works, or if there's any truth to that.
Really interesting question, Brendan. Two things that come to mind that I want to describe. First of all, let me just say about the cortisol by itself. It's interesting, and what we keep on seeing, and it fits the newer literature about that. So cortisol is the stress hormone, one of the stress hormone, but is one that's very well known. It's secreted from the adrenal glands. Um, uh, in response to stress, and it has sort of long lasting effects more than adrenaline. And for years and years, and Dr. Ulu and me came from the Sapolsky labs. Robert Sapolsky is one of the people who did all the seminal work about glucocorticoids, and all of us were steeped in the way of thinking that cortisol is bad. Cortisol is really bad for the brain, and the higher it is, the worse the situation is for the brain. And in more recent years, there's understanding that it's a much more complex situation. And in fact, PTSD patients a lot of times show a blunted response. So there's actually not a big enough rise, if you will, of cort, and the very steep increase in secretion of cort, the big peak of cort secretion, and then timely shutdown of the response is actually the proper response that seemed to be the one that comes with resilience, and not having this response is something that doesn't work so well, or that is associated with more adverse effects afterwards. So that's an interesting thing about cortisol. In fact, it is so accepted now that there are clinical trials, and more so to supplement cort. That when people come in after trauma, and they might actually even get supplementation of the cortisol hormone to help them mount a response and quickly shut it down properly. So that's one thing. Then dopamine comes into play, and that's another interesting story. And you were talking about addiction. And to me, when I hear addiction, I'm thinking about the reward circuitry. So circuits in the brain that are involved in the processing of a reward, and the sensitivity of dopamine. And then there are two types of receptors, dopamine 1 and dopamine 2, that play a role in that. And those are other brain areas. Those are not the brain areas that we've looked at, but brain areas that are connected to the prefrontal cortex. And we've been studying those in the lab for a whole other project. This is a project that we had looking at neural bases of empathy and helping behavior. And we saw very interesting dependencies on activation of cells that have dopamine receptor in those other brain areas. And so now there's a student in the lab who decided to look at the response to stress and those myelin changes in those areas where the dopamine receptor is. But she's asking questions about impulsivity that seem to be very tightly related to those reward circuitry, and asking whether we could see changes in myelin and therefore changes in neural activity in those areas that come from stress in animals that end up mounting an improper impulsive responses. And in fact, I mean, she's probably going to look at impulsive and compulsive responses, both. I don't know. Does that answer your question? That was a bit of a long response.
Oh no, very very interesting. I really appreciate that. Um, thank you.
Thank you so much, Dr. Kaufer, for that. Victoria, do you have any questions?
I do. Thank you. Thank you. And I want to thank you, Dr. Kaufer, for coming to Clubhouse and to Science Society to present your research. It's so exciting. My question is, has there been, do you notice a decrease in myelination when the stressors are removed? Has your research explored that?
Wow, you guys have such good questions. That fantastic question, and the answer is I don't know, and I'm really curious to know. Um, how long term is that now? For that, what we would need to be able to do is longitudinal studies where we're able to. So, for example, with people, you can imagine somebody coming in and having a scan, and then having a scan a few years later on and seeing how that changes with life, with interventions, with just time passing. With the animals, it's a little more complicated because those are endpoints, right? We right now, the way that we have to look at the brain includes taking out the brain, and so you can't follow this up. Um, but that is something that we are interested in doing. So we're trying to figure out ways to follow those animals live so that we can now look at a kinetic follow-up of the development of the response. So let's say an animal is now showing us some more myelin in this brain area. What happens now with time, um, and how that changes? And I would love to know that, but I don't yet.
Thank you so much. It sounds like you're speaking to epigenetics too.
Yes, yes. So, the stress field has been really seeing phenomenal things with epigenetics and things that change, so changes that occur on top of the DNA, right? That then changed gene expression. So this would be one of the places where you would imagine it's perfect to see that kind of response. One thing that we saw that to me is sort of a beginning of a hint towards a very long, prolonged response, but not in everybody, is we've done the stress exposure in peri-adolescence. So around the adolescent time in the animals, we've done this, and you see an immediate response. Then, if you let animals age or mature into their adulthood, and you test them, then you see a long-term effect, but only in the females and not in the males. And so in the male brain, most of the male brains just, they then look not different than their control counterparts, whereas the female brains had a very distinct long-term change in myelin patterns.
That's really disturbing. Thank you. A lot to think about.
Can I
Yeah.
respond to Daniela?
Uh, no, could you please hold on? We're kind of going in PTR order, so just in a moment we'll get to you. I hope that's okay. So, but don't worry, we'll definitely get to you. But the next person in line is Denise. So, Dennis, do you have any questions for Dr. Kaufer?
Thank you, Cece. Dr. Kaufer, thank you for joining us. Really interesting discussion. These questions are adjacent. They're a bit out of the scope of the question of the paper, but I was hoping you might be able to speak to them anyway. So I was curious. We were discussing adolescent versus full-grown. I was wondering if adolescent rats had more susceptibility or less compared to adults. I was also curious if there were any changes in diet that correlated to changes in myelin production. Those are my two questions. Thank you.
Got it. Yeah, good questions. So for the acute stressor, this very short 3-hour exposure to the predator odor, um, overall the adolescent mice seemed more resilient. They had less of a response than the adults did. Um, what we see when we look a few months later, when they're adult animals, which we didn't do in adults. We didn't follow them that long after. We followed them a month after, but we didn't wait the same amount of time. But in adolescent ones where we wait until adulthood, then, as I said, the susceptibility was very much tied to female and not to male rats. So the male rats actually seemed resilient even over time, and the female rats seemed to have a long-lasting change. So that was the answer of that. But if you're just looking at the broad groups overall and the acute response, it is less so in the adolescent than it was in the adult. We are also thinking, but we're not doing it right now. But there's a reason to think that there would be critical windows for plasticity in the system. And for example, myelination, a critical window for that is after birth and the first 2 years of life in humans, and the first 2 months of life or the first 2 weeks of life in rodents. That you can expect that this would be a critical window because this is when myelination actually happens, and it might be with that when you interfere in that time point, then the effect is much bigger. But at the moment we're not studying that. Yeah. And then your other question was about diet, and I think that's really interesting. And there are things that you might think will interfere with that. We have not looked at that, and all of our animals are on the standard lab rat diet, so rodent diet. So I don't know, but good question.
Thank you so much for your answers. And last question, is there any potential to use this to treat active or chronic COVID infections?
Oh, for COVID infections. Um, I would say it's an interesting idea. I don't know. We have not looked at myelin in COVID brains, and maybe others have. And maybe in time it'll be really interesting to look at long COVID and changes in myelination, but I don't know the answer to that.
I appreciate your answers so much. Thank you so much.
Thanks for your questions.
Great questions. According to my PTR, it is Dr. Shaw next.
Thank you so much. So thank you, Daniela. It was very fascinating, presentation. So my question, I'm assuming whatever you just explained to us, it was about hyper excitability. Am I right? Which it can be related with the myelin sheath and action potential, which we know that when we have a myelin sheath, they are actually facilitate the action potential conduction, allowing the axon while eliminating the potassium channels. Am I right?
So in fact, the work that we've done on hyperexcitability is in a different model system. That's our work with the blood-brain barrier. And in this particular project, we haven't yet recorded neurons. So it makes a lot of sense what you're saying. I would expect that we would, with more myelin, that you might actually see increased conduction velocity and
Sure.
therefore maybe hyperexcitability, but we have not recorded, so that I can't really tell you. I can just guess, and
Yeah,
I
it
think your
just
guess is right, but I
yeah,
don't know.
it just came into my mind because you just explained about the. Actually you had a trigger, right? You just stimulate the animal because it was an animal model, and it just came into my mind to ask you this one, and also about the associative memories. I was just wondering, could you find any evidences around this concept as well or not?
Of associated memories. So tell me what you mean by that.
So associative memories, because we are, in some of the, especially about the PTSD, we know that they just experience trauma and they traumatized before. So anytime that they just exposure with the trauma, they just made the associative memories. That's why I was just wondering, because you modeled something like that for the animal. You expose the animal to the trauma somehow. And I was just wondering, could you find any evidences around the associative memories after trauma or not?
Yeah. So you're asking. I think what you're asking would actually require sort of a re-exposure to the same stressor, which we have not done here. What we've done is a fear potentiation that is not related to the initial trauma, but it would be very interesting to try and probe it with a could with a re-exposure to the same initial stressor and seeing whether one can see.
Here we go.
So,
Thank you, Daniela.
yeah.
Yeah, thank you so much, Dr. Kaufer, for that. And Dr. Shah, that was a wonderful question. Thank you. And next, we have Jennifer. Hello, Jennifer. Jennifer, are you there?
She might be away from her phone. So maybe we could circle back to Jennifer. Um,
No, I'm here, but I don't have a question. Thank you.
Oh,
okay, well, glad to have you still,
Thank you.
Jennifer. What about Jihan? Welcome, Jihan, to the stage. Did you have any questions?
I mean, I didn't have any questions, but I had a comment.
yeah, go ahead. Share your comments. Yeah, questions and comments are welcome. If anyone is in the audience wants to come up and share any questions, comments, adding to the discussion, that's also fine. Please come up and share. Go ahead, Jihan.
So my comment was about anybody can have trauma, and I respect that, but there's generational trauma. I'm black. I'm from Africa, so I already have that too. However, also I'm Muslim and I'm a woman, so the generational trauma is about me. So I'm traumatized about all these aspects and all these cultural, economic, also political aspects. So how does that relate to you all? Thank you.
So your question is, how does generational trauma maybe express itself in that way?
Correct. So generational trauma,
Okay.
being from a ethnic minority and a woman, is relatable and non-relatable to all, but relatable to some.
Yeah, thank you, Jihan. That's you're bringing up what I find is one of the most interesting and important topics in stress research these days, and it's the inheritance of trauma, or there's sometimes it's called intergenerational transmission of stress legacies. I've seen it called historical trauma. Sometimes it's really so important, and very very new concept, I think, to the field. And I could say, I'm from the old time, right? I've been around the stress field now for about, I don't know, 20, 30 years. And I've seen how slowly things come around, and people start to kind of accept, when I started, there was this flinching of, oh, mind body connection. And, this is like on the fringe of actual science, of things that we now accept as very well known. So I think the idea of the biological concepts behind the intergenerational trauma is there now. It's just making those moves of trying and starting to get into the front stage and being accepted by the community. But I find that the work there is just phenomenal. And some people like Dr. Brian Diaz from USC, and previously from Emory, and others that just have very very elegant work proving that things have a mark, mostly epigenetically in different ways, that carry across generations. And some of it is done in rodents now, and some of it is done in, even I've seen in flies and in nematodes and C. elegans, but in a way that really precludes a lot of the stories that we have around to obscure it. That so over the years, when it was said, the response was, oh, it's cultural, oh, it's behavioral transmission. But you can't really say that anymore when you're talking about rodents, when you're talking about in vitro fertilization, when the parents are not even ever around the offspring, yet you still see the marks of the transmission of trauma or stress that occurred in previous generation. So I think this is exactly the thing to follow, and I'm eagerly waiting to see that take the place that it should be in our discussions about about society and about medicine and about understanding variability of individuals and their response to stress and where it comes from. So really, thank you for this question.
Yeah, thank you so much, Jihan. That was quite an interesting question. And Dr. Kaufer, that's so great that it actually shows intergenerationally in the brain. And I think that just like opens up a whole new field of research in seeing how some of these things might affect behaviors of successive generations as time goes on. Just, the traumas of the past and like ancestral traumas. So that's definitely really interesting. Jihan, was your question answered?
Kind of.
Okay.
Thank you.
All right. Well, yeah, thanks for that. And finally, we go on to Parth. Hello, Parth. Are you
Uh,
there?
Yes, I'm here. Thank you for the opportunity. First of all, congratulations on the paper, Dr. Kaufer. I'm not a neuroscientist by any means, but let me try to formulate my question because back in grad school, one of the movies we were asked to watch was a movie called Lorenzo's Oil. The movie covered the movie is about this kid who gets adrenoleukodystrophy. It's a condition in which the body fails to metabolize certain fatty acids, which begin destroying the myelin sheath, and it's a seriously debilitating disease. They don't survive beyond a certain age. Now, looking at the title, I haven't read this paper yet, but looking at the title, uh, increased myelin seems to be, while in the case of adrenoleukodystrophy, destruction of the myelin causes disease. In this case, it's the opposite. Increased myelin seems to be kind of some having correlation with anxiety and PTSD. So my question is, I haven't read the paper yet, but is is it just a correlation, or is it an absolute definitive link between increase in myelin and anxiety and PTSD? Thank you very much.
Thanks. Yeah. Good question. So clinically, it's very clear that decreases in myelin are debilitating, and the most common example that people usually know about is multiple sclerosis, where there are lesions in myelin and demyelinating autoimmune disease, and there's immediately functions to that. So it's very clear that our brains needs myelin, and a decrease in that is devastating to the function of the nervous system. Now, what we're showing here is really outside of what you usually see. It's not increase in myelin is not something that one sees a lot. There are, in aging, sometimes people talk about hyperintensities that you might see in myelin, and those probably are not good. But I think the answer is probably that you need the right amount. So too little is definitely not a good thing, but maybe also too much and in the wrong place is not a good thing. Now, is it just a correlation that we're finding, or is it something that is involved in the pathogenesis of the disease, which I think was your second question? That's important to know, and we don't have a great answer yet. We have sort of a beginning of an answer, as I said. So this, taking a viral vector and manipulating and changing the amount of myelin without changing anything else, and then seeing a functional effect of that tells us that it could be that this is part of the disease. But it's really just um, first steps in understanding that we don't know enough yet, and I hope we will in future years we'll know more about that. I can tell you that a lot of times that's how we ask questions. So, for example, I alluded before to the other work in my lab that's looking at changes in the blood-brain barrier. We do what we call gain of function and then loss of function. So if we think that changes in blood-brain barrier are part of what causes aging, then we take a very young brain and we manipulate the blood-brain barrier to be open locally, and then we follow that brain and we see that it ages quickly. And then we manipulate it back and we reduce blood-brain barrier opening, and we see that we now restore a youthful function to that part. And then only when we do all of those things together we can say, yes, I know now this is the pathogenesis, because that is enough to age a brain, and that is required for an aged brain. And so this is, there's a big step still between where we are with the stress project and really understanding the deep details of that. But that's where I hope eventually we'll get to.
Yeah, thank you very much. So a quick follow-up question, if I may. Do I have the permission to just a quick follow-up question? Is it okay?
Yeah, go ahead.
Yeah, thank you. So so to to rephrase what I earlier asked, so I think you've already answered the question, but for okay, can a person diagnosed with PTSD and anxiety not have a regular myelin and still have anxiety and PTSD? It's still possible, right? So anxiety and PTSD could also be not directly linked with just the myelin sheath buildup alone, right? So did I ask the question right?
Yeah, absolutely. So if I generalize from what you're asking and put it in the terms of what we think about things, is you're asking, is it an absolute biomarker? Can I image somebody's brain and just by the amount of myelin say this person has PTSD? Or even more so, can I say this person has PTSD and they specifically will have problems in avoidance? That the answer is absolutely not. This is not a biomarker. A lot of even work that is meant at things that are trying to be a biomarker, it's very, very hard to move from a finding at the level of a group to an individual. Meaning, even if I know that there is a correlation and people, individuals that have more of this would have more of that. Now, if I take measurements of one person, can I say they have PTSD, yes or no? I cannot. And in fact, there is no real good, that I know of, clinical biomarker that somebody could do that they can measure in your brain or in your blood, or even in your brain. Let's say I could measure in your cerebrospinal fluid something. Can I say you have PTSD, yes or no? The answer is no. And most of the psychiatric disorders work like this, that we don't have very good biomarkers that we can actually, we look at an individual and we know yes or not exactly.
Yeah, thank you very much, Dr. Kaufer. I appreciate that. Thank you.
Yeah, thank you so much, Dr. Kaufer, for answering those questions. I just wanted to quickly circle back to Katie, who came in a little later. But Katie, do you have any quick questions or comments to say to Dr. Kaufer before we move on? And then we have Tierney and Linnea, and then yeah.
Gianni and Lena, go ahead, please. Thanks.
Okay, all right. Well, Tierney, hey, go ahead.
Hi, thank you so much for this room. I'm really enjoying this very, very much. I have a quick question-ish, and I'm curious. I understand that vitamin B12 is really supportive with the myelin sheath, and I was just curious if there was any like baseline studies done around B12 levels and the myelin sheath, and also its relationship to when people have really, really low B12, like 200, 400, that they can have increased anxiety and nervousness and things. And I'm just curious, with from a macro micronutrient level, has any of that been kind of looked at, or did you get my question? Sorry, little
I, I,
fragment.
I totally, yeah, no, I completely get your question, and I think it's very interesting, and I do not know the answer to that, but I'm going to look it up afterwards.
Yes, please do. Please, 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 please do. And so, just for a little bit of like information on the backside end, when you were talking about mental health and well-being and stability, I used to support people with from a business with vitamin B12 injections, nutritive IVs, and the spectrum of people that we would have come in. Whether in 2019, people are still getting electric shock treatment for depression and anxiety and nervousness, and they would come to us to counterbalance that. And then people that were bipolar and going into dips, my doctor started asking people to get blood work done around that time, and we're noticing when their macro micronutrient levels dropped, so did all of the other issues kind of started to rise. I'd be more than happy to share information with you about it, but I was just curious.
Yes, thank you. I'm going to look it up. And if you want to send me an email, that would be lovely.
Okay, will do.
Thanks.
Okay, and then next, Linnea. Is it Linnea or Linnae? I'm sorry, I don't want to say it wrong.
Hi there, it is Linnea, so pretty close.
Okay.
Yeah, I haven't been here that long, so I'm sure I missed a lot of good information. But I was just wondering if anybody here knows more about the relationship between like the gut microbiome and myelination, and just assuming like if your gut is in dysbiosis that you'd have increased inflammation and how that might affect the myelin in general.
Yeah, excellent question. So, again, I do not know the answer to that, but I can tell you that we've been thinking about that. My husband is also a biologist and a professor of biology at UC Berkeley, and he studies gut microbiome. And he studies the effects of gut microbiome in aging, and we've kind of started broaching thinking about how to look at that, but he's studying an organism that does not have myelin. So I do think it's a really, really interesting question.
Linnea, I can comment a little bit. We're actually looking at this in one of our studies. It was an intervention study in patients with depression and obesity that got both a lifestyle intervention and problem solving therapy. And we got fecal samples to do gut microbiome analysis as well as neuroimaging, looking at both resting state functional connectivity as well as diffusion weighted imaging to look at white matter integrity. And we were also in this cohort, we measured levels of pro inflammatory cytokines. So it's all kind of association type analysis and correlational, but we can look at to see whether alterations in the gut microbiome are subsequently associated with elevations in pro-inflammatory cytokines and see if those are also linked to impairments in white matter integrity. So we're in the middle of that analysis, but we've looked at other cohorts and looking at relationships between inflammation and white matter integrity, again in the uncinate fasciculus, which connects the areas that Dr. Kaufer was looking at in the hippocampus and the amygdala to the prefrontal cortex, and showing that increases in circulating levels of IL-6 and IL-1 beta were associated with lower levels of white matter integrity in the uncinate fasciculus. So stay tuned. Hopefully we'll have some good results that might at least provide some hints to the answer to your question.
Yeah, awesome. Well, I'm excited to hear about what comes out. Thank you.
Ulu, that's fascinating. Do you guys ever look at blood-brain barrier dysfunction, like DCE MRI?
No, I haven't. But a long time ago, I was interested in looking at these ultra small particles of iron oxide, which cross the blood-brain barrier in areas where there's a sort of weakness or damage. But it turns out that the contrast agent that you could use for MRI scans to look at this had some bad side effects. So I never went through with that study. But if there are new imaging modalities that allow you to look at the integrity of the blood-brain barrier, I'd be very interested. I am collaborating with some folks at Hopkins looking at women with HIV and looking at PET-based markers of neuroinflammation, and I think they are looking at blood-brain barrier integrity, but I don't know if that's based on PET imaging or MRI imaging.
Oh, we got to talk about that. I have some really interesting stuff to show you on.
Okay, well, I've already sent you an email, so I'd love to learn more about it.
Great.
That's
Yeah,
great.
thanks.
Oh no, I was just gonna say, Dr. Kaufer, I think, a lot of people here might want to reach out to you. So if you want to share your email, maybe just put it in your bio or something so maybe they can get, in touch with you after this talk. I mean, that's up to you, but yeah, I think that might be a good idea.
Wait, I'm super happy to do that, but I have no idea how to use this platform and where's my bio or anything. But can you do that for me and put it somewhere that people can see?
Yeah, so
Karina?
let's do that. People can reach out to me, and if Dr. Kaufer is fine with it, I'll give out her email from her work email, and you can reach out to her. So yeah, let's do it that way. I think it's just easier, maybe.
Great, thank you.
That's great. I mean, I must say today has been such a fun room. It was, first of all, fun listening to you talk about the paper, Dr. Coffer, and then the questions, each and every question that was coming out. I was just like, I want to know that. I want to know that too. So this has been really, really fun. Thank you. Thank you for being here today.
Yeah. Yeah, thank you so much for being here. I wanted to ask one question. Did you or are you testing also cognition, if cognition changes in mice with increased myelin? And I'm asking because I did with a colleague of mine a couple of years ago a study with maternal stress and in mice and separation, and then we tested all kinds of behaviors, and the cognitive impairment was really mostly what was significantly different. And then we did the RNA sequencing, and we saw very few really big differences, like significant ones. And one was um, oligodendrocyte upregulation, like genes that are related to oligodendrocytes. That's why I'm asking if you maybe also tested for cognition in those.
Oh, wow, that's interesting. Did you guys publish the paper?
No, we didn't publish. So
No.
so we did the study, and we are still like, we gathered enough data to like write. We wrote an independent grant just for that project now, to keep doing more RNA sequencing and so on. So we are hoping for funding, but for more funding. But yeah.
Well, I really hope you guys get to do it. It sounds really interesting. We've also had data that was generated long ago with a postdoc that's no longer in the lab, but looked at maternal behavior that was linked very tightly to oligodendrocyte accumulation. And in fact, that was something that stayed with them into adulthood. And in those animals, we didn't check cognition, but we've been doing that on the cohorts of animals that go through the blood-brain barrier studies. And so I'm hoping to start to connect those, to connect the dots between those two. I can't imagine that it wouldn't be, especially when it comes in the right point in time, like very early in life, that you would not see changes in cognition, cognitive abilities, or memory.
Yeah, it would be interesting if just acute stress would also, related with the oligodendrocyte differences would also impair cognition for, and then it would be interesting for a longer term study to see if, like, with the maybe in resilient animals that go back to normal, that the oligodendrocytes change and then cognition changes. Would be interesting to know. So
Yeah, that'd be really, really interesting. And thinking about gene expression and transcriptome analysis, we have a paper that we're writing now where we looked at we ran RNA-seq on a lot of animals that had behavioral phenotyping for the response after stress. And the most, like, if I have to choose one line to tell you what the finding is, is that the biggest responses is in the resilient animals. That the resilient animals, it's not a lack of response. There's actually a very big response there that I think confers some protective effects.
Oh, interesting. Yeah, that's really interesting. Yeah, so I'm looking forward. Maybe you'll come back and share more research results in a few months or so. This was a great pleasure having you, and we learned a lot. And yeah, the questions were great. So thank you, everyone that participated, and special thanks to Dr. Kaufer. Please come back anytime you feel to our to our club.
Well, thank you guys so much for inviting me, and the questions were just phenomenal. I have now a page of notes of things I want to look up.
Yeah, so I want to make some announcements for this week. Please come back and join us. We have tomorrow Dr. Ksenia Krasileva, and she looks with computational methods at structural genomics of different pathogens and evolution of pathogens and interactions with the host. So especially in times of a pandemic, it's especially interesting, but I think she is really a great researcher and does great work. So I'm very excited to hear her speak tomorrow, and it will be at 8 p.m. 8 p.m. EST, and then on Friday, because we had like feedback from the audience, we talked about climate change innovations for solutions. So we wanted to continue this discussion and first learn a little bit more about where our power comes from now and where we want to move, like which power generation we want to move to basically um, better our situation regarding climate change, among a lot of others. And then on Saturday, we'll have a philosophy of AI and building the minds of tomorrow. It's kind of a post party of the room we had at 4 p.m. EST with Dr. Michael Levine about his mind's technological approach to minds everywhere. So we have a guest speaker. He will talk about his thesis where he wrote about the future of intelligence and minds, and also ethical questions around it. So yeah, stay tuned, and thank you so much, everyone. We are very honored to have such a great audience and such great guest speaker, Dr. Kaufer. So thank you, everyone.
Thank you so much, everyone. Have a good evening.
Thank you so much. May I just ask a very
Thank
quick
you.
question? I just wanted to say, Professor Kaufer, thank you for such an amazing conversation and such an interesting paper. I know that Katerina offered to forward on your email, but is there anywhere else that we can find you, like on social media or on Twitter or anything like that? Or any videos that we can learn more about your work and your research, because it's really fascinating and obviously people are really interested with all the questions, and we really hope you come back.
Thank you. I'll be delighted to come back, and in fact, I also want to recommend that you guys invite one day the postdoc who now has our own lab that's working on the neural pathway of empathy, because I think that's super interesting findings that you might have an interest in. But okay, so Twitter, there is a Twitter to the lab. There's some account. My students are doing it, so you can look there. And I sent Katerina, there was a piece in the evening news in CBS last week, which I think is a great one that summarizes this story and also interviews the postdoc that worked on the paper and our collaborators from the VA are talking, Linda Chow and Dr. Nayland are talking about their side of it. So that might be a really good way to follow.
Yeah, I just posted the pin. We had it in the beginning when the room started, and now I changed it back. So please look up the news article, and yeah, the Twitter account is at Kaufer Lab, because I added the lab account to the announcement. So yeah, and yeah, we would be very honored to have your postdoc that's opening up her own lab. Maybe I'll email you about it, and you can share with me her email. And yeah, it would be wonderful to have her. That would be a great topic to discuss here. So great. So I'll
Great.
close the room. And yeah, thank you so much, everyone.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you. Bye.